Inside every cell in the human body is a nucleus, and inside every nucleus is all the information needed to create a human being. This information is deoxyribonucleic acid, or better known as DNA. DNA has two complementary strands which contain the nucleotide base pairs A, T, C, and G. Adenine, thymine, cytosine, and guanine are all attached to the sugar phosphate backbone in a particular order to code for specific amino acids which form proteins. Each nucleotide has a complementary base that it always binds to, which allows coding of specific amino acids. A always binds with T, and C always binds with G. This consistency allows each strand of DNA to be used as a template in DNA replication. Replication begins in specific spots in the DNA, which are called origins of replication. An enzyme called helicase unwinds the double helix and separates the two strands from each other. When the helix is unwound, it creates strain on the rest of the intact helix and can cause supercoiling. Supercoiling is when the strands coil up like a telephone cord, which shortens the molecule. Topoisomerase is an enzyme which sits behind the helicase and cuts and repairs the strands to relieve strain and prevent supercoiling. Single-stranded DNA binding proteins prevent the DNA strands from binding together and to other outside molecules.
After the parent strands are separated, RNA primase creates a primer which signals the DNA polymerase to start adding nucleotides to the 3' end. A strand of DNA has a 5' end, where the 5' carbon is exposed to it, and a 3' end, where the 3' carbon is exposed. Nucleotides can only be added to a DNA strand on the 3' end, so this presents a problem because the two strands of DNA are antiparallel, which means that one strand goes from 5' to 3' and the other strand goes from 3' to 5'. This presents a problem with replication, since replication only goes from 5' to 3'. Because of this, each strand is replicated differently. The 5' to 3' strand is called the leading strand because it can be replicated continuously, but the 3' to 5' strand is called the lagging strand because it is replicated in fragments called Okazaki fragments. Okazaki fragments are formed between primers in the 5' to 3' direction. DNA polymerase I then replicates the RNA primer with DNA nucleotides, and then DNA ligase joins the segments together. This process happens over and over on the lagging strand until there is a complete complementary strand. At the end of DNA replication, each new strand has one old strand belonging to the parent helix and one newly formed strand.